Yeah, thank you for inviting me for giving this talk. And I am going to give basically my presentation in two parts. The first part is on the gelatinous macrozooplankton, which is actually work done by Russ Hopcroft and Dougal Lindsay. And then I'll switch over to a PowerPoint that is giving some details on the benthic work. So this first part is a movie that is about 10 minutes long, and I was unable to post it as a movie on the website. So if you're on the phone, I did frame grabs of the movie. So hopefully you will still be able to go along. And so this is about an update on the Chukchi Borderland Ocean Exploration Project, where we went with the icebreaker Healy up to the Chukchi Borderlands in July 2016. And so again, I'm going to start out here, and I'm going to start this little video. And I hope everybody can see this. And I will basically narrate our trip, where of course we had sea ice and polar bears and everything. This is a station map. We did 13 stations, and 10 of which we looked at with an ROV. I'll talk about that more. We had a very stratified water column that you can see here with temperature and salinity sections. And really the tool I want to focus on is the ROV. This is what you see here. And so it has a whole variety. I don't know if you can see my cursor, but I'll still point out some things. A whole variety of different camera settings, and then also sampling tools. There's a snorkel here. You'll see this later a little bit in action, and then some other samplers here. This is the pelagic configuration. We had a bit different configuration for benthic sampling. And this is inside the control van on the back deck of the Healy, where all the controls are, all the cameras. Once the ROV comes back on board with the samples, you can then very carefully remove samples with minimal disturbance, which particularly for this part, the gelatinous zooplankton, jellies in common language, I guess, you can collect them with minimal disturbance. And so it's really important, because these things really disintegrate very easily when caught with nets. So this one I'll get back to later, so I'm actually not going to talk about it much. But so starting out here, this is a siphonophore. This is normally a very common siphonophore. We didn't find it all that often. But it's a very shallow siphonophore. It's called Mertensia ovum. I will show a number of these plots here, and I just stopped the video for a second to explain the plot layout. So what it is, it's during the dive of the ROV, we would make counts, and these are of these individuals of the different species. And so these are counts per minute on the x-axis, and then depth on the y-axis. And so in general, the layout of this video here is that we go from shallow water species to deep water species. The different symbols are different colors and symbols for the different stations. And so it gives you an idea about how common or not common these various species were. This is another siphonophore, also reasonably common. That's baleenops. It has these large oral lobes that are used for capturing zooplankton and things like this. You see, it's a little bit deeper to about 500 meters. And then another siphonophore here, this is Beroe. Beroe is characterized by it has no tentacles. It has sort of a reddish color like this. It is a voracious predator. In a moment on the top, you'll see the mouth coming out. It eats other siphonophores, such as the baleenops and Mertensia. It also was pretty common, also restricted to that other top layer, 500 meters, where its main prey is. This one is a different siphonophore. This is Adora. It is similar to Beroe that we just saw, but it is much more delicate, and it starts to extend a little bit into the deeper waters here. And it also wasn't really quite as common. We didn't see it at many stations. This is another siphonophore that is interesting, because you might have seen it comes up again. The tentacles have these interesting little knops on them that look like pearls on a string. This is Euplocames. And these little pearls on the chain here are actually side branches that coil up, and we'll see a close-up of this in a second. And again, up to about 600 meters depth. So this is a euplocamis where you don't see the tentacles themselves, but these brown coils you see, those are those little balls that you saw earlier in the picture. Now, this is not a ctenophore here. This is a siphonophore, and it's a very small colony, only a few individual polyps actually making up this siphonophore. It is quite common. You see this here, and at a lot of different stations. It's very small. It's only a few millimeters in size. It does occur also in other regions of the Arctic. This one also is a siphonophore. This is Ratchakovia, and it also is a fairly common species that we found at a lot of species. You see a whole clump here of these different colors. And again, to about 800 meters depth, but occasionally we found it also really in deep waters. It's about the size of a pen, or a thick pen, and you can see how the colony has different parts to it. Some parts are more floats, and others are different types of polyps. This is a free polyp that dissociated from the colony, so they also occur in solitary form. And so this was brought into the lab, and so we could study also individual polyps. And we'll later see another siphonophore that has a strong structure to the different polyps. This one is a Hydra Medusa. This is Smithii arctica, distinct by the 8 radial canals and 8 very long, thin tentacles, very common, distributed over a very large depth range, all the way down to about 2,000 meters. And in the next picture here, you'll see how broadly it can spread out its tentacles. So they are actually very good hunters. They sift through a lot of water. This one is Botryonema, and it's sort of a cone-shaped Hydra Medusa, also 8 radial canals. We'll have a little bit of a close-up in a second. Again, a strong or large depth distribution that you can see here, with a few more stations in shallow waters up to about 1,000 meters, but then a number of stations where the distribution also reached down to about 2,000 and deeper. This also is still Botryonema, and what you will see, what is distinct, it has this little knob-like extension at the top of the bell, and that's specific to this species, and it enhances this cone-shaped structure. This one is a ctenophore. This one is Lacortina. It's an undescribed species, and we knew this species from collections in 2005, but we were not able to collect very many, and so we were very excited to collect a whole bunch more of individuals. Well, a whole bunch might be over stating things, but we collected a number more for DNA analyses as well as morphological studies. It's about the size of a grapefruit, also the color of a grapefruit, although its pet name in the lab was Mr. Pumpkin. And it has very, very long tentacles, as you just saw. I stopped the movie here for a moment. We saw it at a number of stations, but not too commonly. But again, we were able to collect a number of individuals. This one is Agena Hyperborea. It's a very clear Hydra Medusa, and again, 4 radial canals, distinct by the dark lining of the stomach and the manubrium and the gonads. And again, this was not a very common species, so we were excited to see it. Just a couple of sightings at a handful of stations here. This is a close-up, and you can see the short tentacle fringe here, and the color of the stomach, gonads, and manubrium here. So one of the benefits of this high let me just pause this for just 1 second. Of the high resolution imagery we can collect with this particular ROV is that it actually allows us to see an incredible amount of detail on these organisms while they're in their natural environment. Once we get them on deck, collected with the ROV is fine, but if you collect them in nets, they are so destroyed that you can't really do anything with them anymore. So moving on here, this is Nectodamus. It's another siphonophore, another very small siphonophore. You see sort of the polyp distinction here inside. In this picture, it is actually attacked by a hyperiot amphipod. It was very rare. We basically had just one station where we had a sighting. So that was very exciting to see and to make some connections. This, in contrast, is a very large siphonophore. You can see here how it's spread out. With the various polyps, and we'll see a close-up in a moment. Again, at a handful of stations, we were able to collect material of this species. And here you see the colony and how some polyps are specialized for the floating part of that whole colony. And then further down, you see great distinction in the various polyps in terms of some are for feeding, some are for reproduction, some are for the defense. And so it is a very, very complex a siphonophore that's up to a foot long. This one is an undescribed species that we knew existed but had not been able to collect any material from ever before. So this was very exciting to be able to collect and get, again, some imagery. As well. So this is a Tallit Anela. This is a hydra medusa, and it's interesting how on the lower side you can actually see how some of the internal organs, again 8 because they're associated with the 8 radial canals, are reflecting the light. The hydra medusa itself is very dark brown. You can also see the tentacle fringe around, but then one tentacle is always leading and is always extended and longer. This is a characteristic of this species. And so here you see the depth distribution. So really we didn't see it above 500 meters. So we're getting really into the deeper species here. This is a Narco Medusa. This is Benthic, and it's characterized by these distinct 4 tentacles that are inserting mid-bell. So you see when the bell is contracting, the tentacles are actually not part of that. It is a deep water species, mostly below 1,500 meters. Although we found it even deeper. And then here you see that there actually are tiny little side tentacles and sensory organs attached to the lower part of the bell as well. This one is an undescribed ctenophore species. When it gets closer, you can see how the stomach and mouth part here are darker brown. It has these two tentacles that can be retracted into the sheaths. And this dark brown lining has given its field name, which is liver lips. I was not the one naming this. It is a not very common species. We were really hoping to see this because, again, previously we were unable to collect anything. So here this time we were. And so being able to get more imagery and actual samples for DNA analysis, that was very, very exciting for us. So this one is Cressona. There are 2 species. This is Cressona norvegica, and you can see how sort of it's opaque, has a very dense fringe of tentacles at the bottom here, and it's a deep water species, and the other species is Cressona miltsai, and you can see how different it is, has very long tentacles, and you can see how they're involved in the movement. Here they are all retracted in, and it's probably one of the more stunning hydromedusa we have, given its color and the iridescence of it, and you can see how the gonads are attached to the radial canals and the red coloration. This one is Benthic, and another quite fascinating and stunning Hydro Medusa, you can see here again, has a lot of iridescent, has longer primary tentacles and a fringe of slow secondary tentacles. This one is a undescribed ctenophore species. It's darker brown, and interesting, this is a benthic ctenophore. That's very unusual. So it can be floating like it is here, and then it spreads out its tentacles, and you see the little side tentacles hanging down here in a curtain. And then when they capture food, they bring them up to the main tentacles and bring them to the mouth. That's how these tentacled ctenophores feed. You can see how elegant and coordinated their movement is. But more often than not, this species is attached to the bottom. It uses one of the tentacles to hold onto the bottom and then just floats there. This is actually collecting it with the ROV, and so we were able to get a number of good samples. This one is the last species. This is Trichogastria. It's a benthic jelly, and this one, again, sits for the most part on the bottom. It has these primary longer tentacles that spread out on the surface, and it's for prey capture. And then these lower, smaller tentacles on the bottom are adhesive tentacles, and it uses those to attach to the bottom. This is a species that's common in the Arctic as well as the Antarctic. So this was the part about the jellies, and oh, sorry, it starts all over again. That was not on purpose. So I will just switch over and keep going right away with the part on the benthic investigations we did. And I'd like to acknowledge my co-authors, Sarah Hardy and Brenda Norcross here from UAF, and then Bodil Blum and Irina Zulai, who is a PhD student, and she participated in the cruise, and they are from the Arctic University of Norway. So for benthic studies, we used a box cora, trawls, and also the ROV. And for the purpose of this update, I decided to stick with the theme and really just show you updates from the ROV. So here we did, again, benthic ROV dives at 10 stations, and at some stations we had a chance to do more than one dive. I will focus mostly on benthic invertebrates, although we also had fish, and I mentioned them here and there, but I'll focus on the invertebrates. So in terms of number of taxa that we found from ROV imagery, identified from the imagery, about 75 species, and you can see here most of them within the Cnidaria, such as this anemone here, and echinoderms such as this sea star over here. We can also look at them at the species composition in terms of stations. And so,
Katrin?
Yes?
I don't think that we can see, right now we're just seeing a polar bear.
Oh, sorry. I'll see if I can change that.
So you'll probably have to click share screen again and then choose the video that you want.
It doesn't come up, my share screen.
Let
me close this down. Oh, now we're back. Okay, sorry about that. Here we go. Okay, should I go back real quick? Maybe at least to this slide here. Hold on, go there. Can you all see this now?
Yes.
Okay, so I was starting to talk about the number of taxa that we find, and so most of them are within the Cnidaria, this anemone, or the echinoderms like this sea star here. And then now looking at the distribution of taxa with station, these are the stations, and they are ordered by depth. So this station 6 was the shallowest station at 500 meters, and station 7 was the deepest station at 2,640 meters, and then everything else in between. And you can see that there are more species at the shallower stations than at the deeper stations. Sort of a break happening right around here with this station 2, which was at about 1,100 meters, and then station 11 at about 1,800 meters. So there was a bit of a break there. And we see this also if we look at which type of taxa are occurring in these stations. This is the same ordering of stations here. And you can see that in these shallower stations, the contribution of, let's say, in blue echinoderms and in red cnidarians, so you have the legend here, is pretty similar. And then when we get to these deeper stations, that really changes, and echinoderms as well as cnidarians get a little bit less, but we have an increase in sponges in purple here, and in some of the arthropods, such as this shrimp that you see in the back here. So we've collected a number of these deep water sponges, and we're still working on identifications. It's a notoriously difficult group to work with. So currently we're still going by field names such as a lollipop sponge, this one here, or a golf ball sponge. So you can get a little creative about naming things for field names. We also did a cluster analysis where you take all this information and you order them, the different stations, the community at these different stations by similarity. And again, we get a pretty distinct breakout of these deeper stations to the left group here and the shallower stations to the right here, with the red indicating that there's no statistical significance between or in the community composition among those stations. So depth really is playing a role here, and it's a fairly distinct break that we're finding there. So then I decided to maybe focus on some special benthic special features. One of the special features we found are drop stones. So most of the environment is soft sediment, but once in a while you find these drop stones. This may be about a foot or two sort of in size. And then all of a sudden you find all kinds of things like this very clear ascidian, these little scallops here, hyalopecten, a number of different anemones, octocorals, polychaete tubes. So there's this accumulation of high diversity at these drop stones. The other feature we looked at, and we specifically, there was an objective of this project, was to look at Popmarks. So we knew from previous work by Larry Meyer that there are these Popmarks fields which are extinct gas hydrates that are nowadays, from all we can tell, are not active anymore, but they leave these distinct depressions in the seafloor. Here you see a depth contour that was taken during an ROV dive, and you can see we go down and reach the bottom, and then this first part here was within a Popmark, and then we would go outside of a Popmark, and the depth difference is about 50-60 meters to the surrounding plateau. And so we were interested, is the community inside any different than outside? And so down here were a couple of multi-beam pictures of these depressions, on the left picture here in blue, and on the right one in green. Those are those depressions. So looking at a couple of examples, it was really difficult to position the ship right above one of these Popmarks because, as I said, they're not that large. We have strong currents and everything. So our first attempt wasn't all that successful, but I still wanted to show you because we found some interesting critters here. So an Arctic skate, a fairly large fish, was fascinating to see. We found stalked crinoids here, Benthic crinus, which are really restricted to the deep sea, and we found a lot of them, so that was interesting. These neon yellow anemones, things like this. But really what I want to point out to you is this particular brittle star. And I have a little movie clip here. So look at the lower right part where you might be able to see this brittle star, and it is a swimming brittle star. And this was not just because we came and disturbed it. There were lots of them, and they were all swimming. They can eventually stop and then they sink back down, but they do swim on their own, perfect breaststroke, and was really fascinating to see. Here, another station, again, where we had plateau, and then later on, we went into the Popmark, and in the surrounding plateau, we found eel pouts, more of these stalked crinoids, anemones, and brittle stars. That was sort of the dominant parts of the of the community. But then when we went into the Popmark, we basically, the only thing we found were anemones. And I have another little movie clip here that shows you how it was within that Popmark. And you can see how there's sort of this meadow of these anemones, and pretty much more or less nothing else. And it just kept going like this. It was really fascinating to see. The last one here was a little bit deeper. The other two were about 500 meters. This one was 940 meters. And so we were able to actually really go right through this Popmark. We found lots of gooseneck barnacles. We found them within and outside of the Popmark. They're a little unusual because, again, they need a hard substrate to attach to. We found this brittle star, which is Ophioscolex. And that is a brittle star that really, the moment you touch it, you bring it up to the surface, it completely disintegrates. It's like slimy, and it completely falls apart. From all I can say, I believe this might be the only picture of a completely intact Ophioscolex that exists, because it's really incredibly difficult to find them and to take pictures of them. But both of these were inside and outside of the Popmarks. Unique to Popmarks were these very large pycnogonids, sea spiders, colossendies, and these are very large. And again, I have a small little movie clip here. I promise you it is moving, although they are very, very small, slow movers. And they have this, you know, prehistoric look to them. They are predators. They bore their proboscis into the prey. They do prey a lot on, for example, anemones, which are common around there. But these pycnogonids were really restricted to the Popmark, and we had fields where we would see 10 of them at the same time. They are probably a half a foot wide, so they are very, very large individuals. So just in case, in terms of a summary, we have found a number of new records. This swimming brittle star I showed you a movie on, Ophiostriatus striatus. It's a new distribution record. It's known from the European Arctic, was not known from the Pacific Arctic. It is one of only 6 species of the about more than 2,000 known brittle star species that can swim. So that's a very interesting behavioral thing. This gorgeous scale worm here is in the genus of Bathyphovelia. Currently, there are only 2 species described for this genus. We're not sure yet if it is one of them or possibly a new species. This little scallop I showed you on a dropstone before, a major geographic extension from the European Arctic, and again, potentially this might be a new species. And then also another clam or little bivalve here, Bathyarca, again a major geographic extension into the Arctic, wasn't known from the Arctic before, just from North Pacific and other lower latitudes. And again, we're not sure it's that species. It again might prove to be a new species. And with that, with a little smiley face laid out on the back deck of the Healy, I take any questions that you might have. And I hope I stayed within my time limit.
Yes, you did. Thank you so much, Katrin. Are there any questions for Katrin?
Katrin, maybe you could identify sort of where the stations were. Do you have a map of where they were?
I do. Oops. You mean the Popmark stations?
Sure. Where overall in the Arctic geography are we?
Oh, I'm so sorry. That was part of the movie clip, so I didn't repeat it here. So I don't have actually a map unless I bring the movie back up. But it is off the north of the Chukchi Shelf. So basically, if you were at Wrangel Island and you would move northwards, that's when you get, it's a transition area between the Chukchi Sea Shelf and then going into the Canada Basin.
Thanks, Katrin.
Any other questions? If not, thanks again, Katrin. Yeah,
I've got one. Katrin, this is Will Ambrose. How did you quantify the benthic counts? Was that just like you did in the the water column? Was that counts per minute of tow time, or did you have lasers on the bottom? How did you do that?
Yeah, we started out with lasers, and then after, I think in our third dive, the lasers failed. So that is still something we are still working on. Currently, what I showed you, number of taxa, is just number of all identifiable taxa that we could find. That's not quantified by area. We're still working on trying to get a good idea of how to quantify it with the lack of the lasers. We do have, as I said, we do have some from the first few ROV dives we did, but there was no way that the ROV techs could fix them. So we were left without them. The problem is that the ROV itself is obviously going up and down, so it's not a specific height that you keep all the time. And the depth recording we have are associated with the clump weight, not with the ROV itself. And so we're still working on maybe using animals that we know the size of as a scale bar. It's quite an undertaking, but we're still working on that.
Now, I was going to suggest that approach. You could always just go to counts per per minute of trawl time if you're at a more or less constant.
We're exploring that as well, but currently we're looking into if we can actually use known size of individuals or of species and use that as a scale bar.
Yeah, good luck. I've done that too.
Yes. Thanks.
Thanks.
This is Natalie. I have a question.
Yep.
Did you see any emission coming out of the Popmarks?
No, we did not. We really looked for that, and we did not see a single a gas bubble coming up. What we did see was a fair amount of whitish sediment, and that we believe are bacterial accumulations that are oftentimes associated with chemosynthetic activity. And we were able to collect some of that sediment, and we gave it to Eric Collins to have a look at it. But as far as I understand, he hasn't looked at them yet.
Yeah, it could be also some calcium carbonate.
Yeah.
Okay, thank you.
Yep.
Other questions?
I have a sort of bizarre question, which is you noted the drop rocks that had a lot of biodiversity around them. Are there other sort of benthic surprises in terms of, I mean, I'm a whale guy, so I think about whale falls or any other sort of surprises you found down there that provided additional habitat?
Yeah, I actually deleted it from my presentation because I thought I was going to run out of time. The one thing we saw were big holes, basically, that were in the bottom, and they are way too large to be made really. Oops, somebody talking. They were way too large to be actually produced by any of the organisms that we saw. They were probably 2 feet wide and 1 foot high, and they were like caves. And so we believe that they're probably physically made by some seized gas activity, but they now do provide habitat. So we find little polyps that are hanging off the arch on the top of these caves, and we find a small shrimp that are moving in and out. So there were a number of these kind of things. We also found a couple of other organisms that I didn't show you here that were surprises, like a very, very long, what we assume to be some type of a coral, octocoral probably, that when we tried to collect it, it was maybe, I don't know, maybe 1/2 foot tall when we saw it. We were all excited, and then we tried to suck it in with the ROV, and it stretched out to probably 10 feet length. So we had some of these kind of things happening.
Wow, thank you.